Vanakkam. Welcome to this course on classics in neuroscience. From this video onwards, we will start looking at a new topic, just neural circuits, specifically circuits of the spinal cord, retina and some invertebrate systems. Shown here is suppose a 3D rendering of a neuron with dendrites and axons, relatively complex architecture, complex network of a set of neurons perhaps with complex dendrites. This picture. In this video, we will look at the background for the rest of the chapter, the rest of the set of videos, the spinal reflex pathway and Renshaw inhibition and muscle spindles. We have already looked at receptor potentials and muscle spindles. Remember our earlier discussion in crayfish research based on that, but here we continue that discussion and discuss about the physiology in a bit more depth and who did the work. So, from individual intracellular recordings, there was an interest in understanding circuits, pathways and systems behavior. So, there was an interest in pathways and systems. Rather than understanding just one cell behavior, because for example, in the human brain you are looking at billions. So, how do the pathways work is of greater interest than understanding the function of a single neuron, for example. So, the earliest work started from the lab of Eccles, of course, in spinal cord, where they studied excitation and inhibition in spinal cord, and they identified the Renshaw cell. This Renshaw cell happens to be a very nice model of lateral and recurrent inhibition. Then came the work in the visual system about how contrast is perceived, for example, and how there is both excitation and inhibition acting together. So, there is a balance between excitation and inhibition in the visual system, in the retinal system. This work was of course led by Granit, Kuffler, Stephen Kuffler, the same Stephen Kuffler, Hartline and Horace Barlow. A series of work in this system. And then we will also look at the extension of the concept of reflexes, command neurons and the concept of CPG, central pattern generators. This will be the overall organization of the set of videos starting from now, but this will be presented in a series of videos, not just this video. In this video, we will restrict our attention to Renshaw inhibition and muscle spindles. Okay. Of course, there was this breakthrough of the use of small or microelectrode in the 1950s. This helped people to study individual neurons and the connections of the individual neurons. They were also interested in studying how neurons talk to each other. How do they talk to each other? So far, in the most general case, they communicate with each other through chemical synapses. That is what we have seen. Intracellular recordings in the brain was a relatively challenging task because of heartbeat and pulsations in the brain. But more than that, the size also matters. The size of the neurons, many neurons within the brain are interneurons. I have already mentioned this, the largest number of neurons, largest set of neurons within the brain, more than half the number of neurons are actually found in one nucleus of the brain, which is the cerebellum. So, these are the granule cells. Almost all of these granule cells are interneurons. These are really small neurons. It is extraordinarily difficult to measure from individual granule cells when there are millions of these. The idea then was to use individual microelectrode to record from the extracellular space of the neuron, not the intracellular recording, but rather you record from the extracellular space of the neuron. This is okay, except that you do not know from where you are recording in the neuron. Are you recording from the cell body, are you recording from the axon, are you recording from the dendrite? That is one. Also, you cannot be sure of the specificity. If it is densely connected, then it might be very difficult for you to identify which neuron you are even recording from. So, this idea of recording from extracellular space from neurons came to be called as single unit recordings. We have seen this previously, but again just reminding you, these came to be called as single unit recordings. There were two major goals or two major philosophies of experimental approach that emerged. One is focused on mapping neuronal connections, that is, what is the output of one neuron and where is it connecting to, what is the output of that neuron and where is it connecting to, and so on and so forth. This is used to mapping, so that you can have an idea of the input output relationships through a map, through the connecting behavior. The other is to characterize the responses of neurons to physiological stimuli, not necessarily electrical stimuli, but physiological stimuli. If it is a visual system neuron, then you stimulate it with light, then see how it is responding, how that neuron is responding. You record from that neuron the response to physiological stimuli. So, this way you get to know the input output relationship. For what input, what is the output for this particular neuron, then you measure from another neuron for the same input, what is the output, then you change the inputs. So, you have a whole set of inputs and you have a whole set of outputs from different neurons. Then you get an idea of, you try to characterize the input output relationship for that nucleus or for that set of neurons from which you recorded, for example. Remember, these are two different approaches. In one case you are not stimulating, you are only recording the natural behavior and the pathway. In the other case you are giving stimulus, not electrical stimulus but rather physiological stimulus, and then recording the response, and I am collating I am making the input output relationships. Both of these worked in tandem with each other, both of them worked together in the 1950s, that led to significant breakthroughs in understanding the central nervous system. This in the 1950s, 1960s, this contributed to improving our understanding of the central nervous system. Shown here is the connections between alpha motor neurons and the muscle fibers and the Renshaw cells, the interneuron that inhibits the alpha motor neuron, for example. We will discuss this function in later videos. Now, we will look at muscle spindles. I will refer you to my other course on neuroscience of human movements for detailed video of physiology of how muscle spindle works, for videos on muscle spindles. Alpha gamma coactivation, that is also on those intrafusal, extrafusal fibers. For 2 or 3 videos my TS will share with you the YouTube links, both in forums, and it will also be available in the description of this video. So, I request you to please do watch to understand the physiology, the neurophysiology behind muscle spindle function in depth. Here I will focus on the work that revealed this function. In 1940s, Lars Leksell, medical professional in Karolinska Hospital in Stockholm in Sweden, started working on a problem that was suggested by Eccles and Sherrington in the 1930s. Eccles and Sherrington observed that there were a set of thin fibers in the motor nerves, much thinner than the other set of fibers, the large diameter motor neurons. If you stimulate these thin fibers individually or in a group, they did not cause any noticeable contraction in the muscle. How is this even possible? You are stimulating a motor neuron, but there is no change in the muscle to which it is innervating. It is innervating in a muscle and you are stimulating it, it is active, but there is no force that is getting produced, there is no visible, detectable or noticeable contraction that you can observe. How is this possible? If it is not causing any contraction, what is it doing? What is their function? It is a mystery. What Lars Leksell set out to do was to study how muscle spindles functioned. I will come back to the previous question in a bit, please be patient. So, remember muscle spindles, we mentioned this in our previous discussion on the crayfish and the receptor potential. Muscle spindles are those that detect length and velocity change, length of shortening and velocity of shortening. These are of two types, 1A and 2 afferents. It was found that these are a different type of fibers. These fibers are called as intrafusal fibers. So, these are modified muscle fibers. The original muscle fibers that produce force is called as extrafusal fibers. These are the force producing fibers. The intrafusal fibers are modified forms of the extrafusal fibers within the long fusiform capsules within the muscle. So, they are in parallel with the extrafusal fibers. So, you are having these force producing fibers and within these you have these small intrafusal fibers. These muscle spindles have sensory axons. So, when the muscle is being stretched, these intrafusal fibers will get stretched and the sensory neurons attached to the muscle spindles to these intrafusal fibers will detect the change in the length or will detect the stretch and will carry this sensory information to the spinal cord and to the brain. Extraordinarily crucial function of proprioception. Do check this out. Proprioception. Crucial function, extraordinarily crucial function of proprioception. So, they sense the amount of stretch and also in a sense the velocity of shortening or velocity of stretching. So, this gives the crucial information about the state of the muscle length, how stretched or how shortened the muscle is. This is what it was giving. It turns out that the central regions of the intrafusal fibers are sensitive to stretch, whereas the edges or the ends like here and here are innervated. The ends or the polar regions are innervated with these thin motor neurons. Remember, there is the set of thin motor neurons which if you activate does not cause any forces, is what we saw. These are the extrafusal fibers and these are the intrafusal fibers, and the thin motor neurons here marked as gamma here innervates on the polar region or the end and they slightly stretch the intrafusal fibers when the muscle is contracting, thus keeping the central region, which is sensitive to stretch, tight all the time. It is always maintained to be sensitive. It is an incredible physiological function if you look at it, because you are changing the sensitivity by stretching. Incredible, unbelievable when you first read about this. For me, when I first read this part way back in 2006-07, when I read about this in Candle and Schwartz, I was stunned, I was completely stunned. This physiology, in my opinion, this was the most fascinating concept ever that I have studied in neurophysiology, but I have not studied the entire Candle and Schwartz, do remember this. So, whatever I have studied, this concept of how the muscle spindles function and how the alpha gamma coactivation causes the stretching in the intrafusal fibers is the most fascinating concept for me. Do check out the videos that I have referred, my TS will share with you. So, this is how the muscle spindles function. This was identified by Lars Lexell. So, remember, so those thin fibers, we left that question unaddressed. The thin neurons that innervate the central intrafusal fibers cause stretching of their ends so as to maintain the sensitivity of the muscle spindles, which are essentially sensors that give afferent information, because it attaches to the 1A afferent and the 2 afferent. This is attaching to the sensory neurons. So, they inform the state of the muscle to the central nervous system, maybe the spinal cord or the brain. Lars Lexell did not pursue this line of work after that. He continued with other things. He was a busy neurosurgeon. He started working on radiosurgery, stereotaxic surgery and other such things that perhaps excited him at that point in time. This line of work was then carried on by other scientists, for example, Stephen Kuffler, Carlton Hunt and other colleagues, where they gave shocks to the fine fibers, and they found that it elicited repetitive firing of the sensory axons from the muscle spindles, showing that the output of the muscle spindles not only depend on the state of the muscle, but also on the sensitivity change that is given by the motor neurons. This set of motor neurons that attach to the ends of the intrafusal fibers are called as gamma motor neurons. The spindle is sensitive to both the amount of stretch and the activation level of the gamma motor neurons. It is a product of the two. So, in other words, you are essentially changing the gain. Your gamma motor neuron activation level is more like a gain. You are changing the sensitivity. So, if you are working with electronics, for example, you know what it means to have a gain knob. If you turn the gain knob, then you see a great change in the output depending on how much you turn the gain knob. It can be compared, you can relate the activation level of the gamma motor neurons to more of a sensitivity knob or a gain knob, for example. Whereas the larger extrafusal fibers, these are not larger muscles but rather larger muscle fibers, are controlled by the thicker or the larger diameter alpha motor neurons. So, there are these alpha motor neurons which innervate the extrafusal fibers that produce force. Then there are these gamma motor neurons which innervate the intrafusal fibers and stretch them at the ends to make them sensitive, to make their central part sensitive. This. So, sensitivity is controlled by the thinner, lower, smaller diameter gamma motor neurons. There is the concept of alpha gamma coactivation. There is also one more concept whereby the alpha motor neuron itself innervates the intrafusal fibers. This is also called as the beta skeletomotor system. I think my students will also share this video with you. Skeletomotor system, whereby the alpha motor neuron, instead of the gamma motor neuron, it is alpha motor neuron that innervates the ends of the intrafusal fibers. That also happens. Almost always when the alpha motor neuron fires, the gamma motor neuron also fires. That way, when the muscle is contracting, the spindle is kept tight and sensitive enough. Please do read about this. Please do watch my referred videos. With this, we come to the end of this video. In this video, we set the background for Renshaw inhibition and spinal reflex pathways, and we looked at the work of Lars Lexen in identifying the function of the intrafusal fibers and gamma motor neurons and muscle spindles. Thank you very much for your attention.